This simple and inexpensive method that we call MDR allows you to isolate enriched populations of Pachytene spermatocytes, round spermatids, and elongating spermatids from mouse testes. The main advantage of the MDR method is its simplicity. You need only standard laboratory equipment that are available in most biomedical research laboratories.
The MDR protocol is great for researchers doing functional studies on male germ cells. For example, groups that are studying spermatogenesis, factors affecting male fertility, as well as paternal epigenetic inheritance.
Minimal starting material is required for the MDR method, and in fact we routinely obtain good amount of cells using just one adult male mouse.
Begin by setting up the necessary equipment and reagents. Set a water bath to 37 degrees Celsius and a cell culture incubator to 34 degrees Celsius, 5% carbon dioxide, and 95% humidity. Place a tube rotator inside the incubator. Prepare and label the appropriate amount of microscopy glass slides. Then draw a ring of about 1 centimeter in diameter with a grease pen and let the grease dry. When ready to dissect the animal, spray the ventral abdomen of the mouse with 70% ethanol and use scissors to make a V-shaped opening in the abdominopelvic cavity. Pull on the epidid.